Hello, grade 10 students. In this presentation and this video, we're going to talk about our new lesson, which is about transgenesis and transcription and translation process. Transgenesis, transcription, and translation. Let's start step by step with this lesson. What the meaning of transgenesis? Transgenesis is a process where a gene is transferred from one species, for example, from a fish to a mouse, from a mouse to a dog, from a dog to a cat, from a human to mice, for example. So this is a process by which a gene is transferred from one species to another, and this gene is expressed in the later. For example, I have an insulin gene is transferred from my body to the mouse body. So this gene is expressed in my body. I'm going to transfer it to the mouse. So it will be expressed in the mouse. So this process is called transgenesis. Transgenic. What the meaning of the term? Transgenic. Transgenic is an organism that carries and expresses the gene from another species. For example, as I said, I have an insulin gene. This insulin gene is transferred from my body as a human to the mouse body. This gene is expressed in the mouse. So the mouse is said to be a transgenic. Transgenic, why? Because it carries a gene from another species and express this gene. Let's move to this experiment. In this experiment, we have the mouse. We remove the egg from the mouse, and then we inject the human insulin gene, as you see here, in the form of DNA into the male pronucleus, and they are fused together with the female pronucleus, and then we transfer a number of altered embryos into the uterus of the foster mother. As we said, the foster mother is we use only the uterus of this mother. The result was the birth of 3 mice. 2 of them, they showed negative result, meaning that there is no insertion of the DNA fragment, meaning that there is no expression of the human insulin gene. Whereas 1 of these mice showed a positive result. It showed the human insulin gene. This mouse is said to be a transgenic. Why it's called transgenic? Because it expressed a gene from another species. And this gene is the human insulin gene in this case. Description for this experiment. As you know, described should be in the form of the passive. So the eggs are removed from the mouse, then the human insulin gene in the form of DNA is injected into the male pronucleus of the mouse, and then it's injected into the female pronucleus. The number of altered mouse or altered embryos are transferred into the uterus of the foster mother after the birth of the 3 mice and the search for the human insulin gene. We have 2 negative results and 1 positive result. So 2 negative results meaning that there is no insertion of the DNA fragment coding for the insulin gene, and 1 mouse showed a positive result, meaning that it's qualified as transgenic. Interpret, it includes analyze plus indication, as you know. So after injection of the male pronucleus of the mouse previously injected by the human insulin gene in the form of DNA into a female mouse pronucleus and transferring the resulting embryos into the foster mother, the result was the birth of 2 mice with negative and 1 mouse which said to be positive result, which is transgenic. So what does this indicate? This indicates that the human insulin gene in the form of DNA would code for the production of the human insulin hormone, where in the positive mouse, in the positive result in the transgenic one. So we can deduce that as a conclusion, the gene made up of DNA code for the production of specific proteins as the insulin one. Okay? You have to know that chromosomes at metaphase, as we said before in the session about mitosis and meiosis, it's made up of 2 chromatids. The substance that made up the chromosome is called the chromatid. Chromatid is divided into either DNA composed of nucleotides. So pay attention here in this slide. Nucleotides, they are A, T, C, and G. They have names, but I don't want to move into the names of these nucleotides. Every one of these is coded in nucleotide. So A is coded in nucleotide, T is coded in nucleotide, C is coded in nucleotide, and G also is coded in nucleotide. Every three together, they are called codon, C-O-D-O-N, codon. Every three, they are called codon or they are called triplets. For example, I have A, T, and C. So the DNA sequence. So this A, T, and C, one after the other. These three together, they are classified as codon. A and T, they are complementary. As you know, the DNA is double-stranded. So if you have in the first strand A, the opposite complementary, it's T. I have C, the complementary, it's G, and so on. So A and T, they are complementary. C and G, they are complementary. Whereas the proteins, they are composed of histones and non-histones. What I'm working on in this session is about the DNA. DNA, by the process of transcription. By the process of transcription, DNA is moving to the mRNA, messenger RNA. And the messenger RNA, by the process of translation, will move to the polypeptide, the amino acid chain of the amino acids. So we have two processes here. The first process is transcription, and the second process is translation. So these two processes, transcription and translation, they are very important in the movement from the DNA to the amino acid or the chain of the amino acid polypeptide or the protein. The DNA, by the process of transcription, gives mRNA, and mRNA, by the process of translation, it gives polypeptide. Let's move now to the single steps in this. First, let's move to the first process, which is the transcription. Transcription process. It's the process where the mRNA is produced and it's based on the given sequence of the DNA. It occurs where? Inside the nucleus. What will happen for the produced mRNA? What is the phase of the produced mRNA? The produced mRNA undergoes mutation, and then mutation means that a change in the sequence. We'll go to talk about this next year, inshallah. So this produced mRNA undergoes mutation, and it's transported from the nucleus toward the cytoplasm. So as we said, DNA to the mRNA means the process is called transcription, but it can't occur without the help of specific enzymes. These enzymes, they are called helicase and RNA polymerase. Let's move to this. Considering the sequence of the DNA molecule shown here, this is the sequence. I have GCA, TGC, ATC, TGC, CAT, and TG. You're going to ask me, for example, why there is only TG. They are all of them. They are 3, 3, 3, 3. Only this. This is a sequence. We take a portion of this. This may be, for example, a sequence that includes 10,000, 100,000, 1 million of the nucleotides. So as I said, every one of these is called nucleotide. And the G is complementary, C is G, A is T, T is A, and so on. Every three, they are called codon. Every one is called nucleotide. So this is the non-template. Pay attention, if you watched the video before, non-template is called non-transcribed also, and it's called the coding. While the other strand, if I gave you the template, it's the transcribed, it's the non-coding. So if you have to find out the sequence of the mRNA molecule, the mRNA molecule is similar to the coding. It's similar to the non-template, which is the non-transcribed, which is called the coding. It's very similar, but you have only one change. If I have read the sequence of the mRNA molecule, it's the same as the non-template. You have to know this. Non-template, it's called non-transcribed, it's the coding. While the other strand, if I gave you the template, template is called the transcribed, it's called the non-coding. That's the difference. We're going to determine this. First, this is the sequence, it's non-template. Non-template, I said non-transcribed, it's the coding. So the sequence of the mRNA, it's identical to the sequence of the non-template, which is also the non-transcribed, which is the coding. It's identical, but We have only a slight difference. All of them, they are same. C, G, C, G, and A, they are the same. But the T is going to be replaced by U. For example, if this is the non-template, the sequence of the mRNA GCA, you have to put GCA. TGC, all of them, they are the same, but T should be replaced by U. U is called uracil. So T should be replaced by U. So this is going to be TGC, is going to be UGC. ATC, A and C, they are the same, but T should be replaced by U. TGC, UGC. CAT is going to be CAU. TG is going to be UG. How may you relate the mRNA molecule to the template to the non-template? Non-template is identical, but T is replaced by U. What about if I gave you the template? Template strand, it's called the transcribe, is the non-coding. So if you have to calculate or to find out, sorry, the mRNA molecule, the template, it should be complementary. So we're going to move to an example about the template. So this is the non-template. This is the template. As you see here, the template and the non-template, they are complementary. G, the complementary is C. This is not required in the question, but I solved this in order for you to know how can we calculate, find out the template if you have the non-template. So here, G, the complementary is C. C, the complementary is G. A, the complementary is T. T, the complementary is A. G is the complementary is C. C, the complementary is G. A is complementary is T. T, the complementary is A. C is complementary is G. T, the complementary is A. G is the complementary is C. C, the complementary is G. C, the complementary is G. A, T, T, A, T, A, G, C. So, one of you is going to ask me a question. Can we find the sequence of the mRNA using the template? Yes. How? It's complementary to the template strand. So it's complementary. How? The C, what is this complementary of C? It's the G. Complementary of C, G, sorry, it's C. Complementary of T, it's A. Here, normal. The complementary of A, it's T. As I said, in the mRNA, you should not have T. So instead of T, you have to put U. The complementary of C, G. The complementary of G, it's C. Complementary of T, A. Complementary of A, it's U. The complementary of G, it's C, and so on. Here, I solved only the first three codons. So as you said, the mRNA strand is complementary to the template. So you have to make it complementary. A complementary is T, T complementary is U. But the mRNA, A complementary is not T. You should not have a complementary as T in the mRNA. T is not found in the mRNA. So T should be replaced by U. If it's complementary with the template, A complementary is U. If it's in the non-template, it's the same, but T is replaced by U. Okay? Helicase and RNA polymerase. What do? Helicase unwinds the DNA of the gene coding for the gene. It unwinds, it make it further, it may move the two strands away. It's called helicase. And RNA polymerase, it synthesizes or it produces or it gives the complementary strand for the transcribed one. The second step, as you know here, I said the first step is called transcription. So the transcription is the first step in the process of moving from DNA to mRNA. But we have to move from polypeptide to the protein. So this by the process of translation. So translation is the process by which the RNA in the cytoplasm can code for production of proteins. So amino acids, the chain of amino acids less than 52 amino acids is called polypeptide. More than 52 amino acids is great than, if you remember, to the protein. What happens for the protein? Some of them, they remain inside the cell, while others, they move and they are exported out of the cell. This is a very important application. Given the following coding, what the meaning of coding is the non-transcribed, it's the non-template, it's the coding. They said give the sequence of the transcribed. Transcribed is the template, it's the non-coding. So it should be complementary. What is complementary of A? It's T. Complementary of C, it's G. Complementary of G, it's C. You don't have to confuse. Complementary strands at the level of DNA, T is found normally. But whenever you move to the RNA, it differs. Okay? So you have to be careful. Transcribed is going to be, as I said, is going to be the sequence complementary. Specify the sequence of the mRNA, and you have to use the genetic code table to give the sequence of it. Let's move step by step. Okay, step by step. Coding strand. The complementary of T is A, so A. Complementary of G is C, so C. Complementary of C is G. Complementary of G is C. Complementary of T is A, T is A is T, G is C is G. mRNA sequence. mRNA sequence, as I said, it's identical to the non-transcribed, to the non-template, to the coding. So this is the coding one. It should be identical, but the T is replaced by U. So A, C, G, CAA. We don't have T. Here we have T. In this code, and the last one, T, C, G. C and G, they are the same. T is replaced by U. Any one of you is going to ask me, for example, if you want to use the transcribed strand, template the non-coding, what can we do? We can do the complementary one. So here, T, the complementary of T is A. T, the complementary is A. G, the complementary of G is C. So C. G, CAA. One is going to ask me, sir, you said you don't have to put T. Yes, this is in the complementary. Here, A, what's the complementary of A? It's T, but we don't have to put T. So it's going to be U. G. mRNA sequence, as I said, is the same as the one for the coding, but T is replaced by U. How can we determine the sequence of the amino acid? Sequence of amino acid, you don't have to study them. But you have to study only 3 of them. And I'm going to tell you which these three. The sequence is going to be AAA is going to do for LYS, CCC for pro, GGG for GLEP, CAA for GLN, and UCG for SER. How can we know this? Let's move step by step for this. Step by step for this. You have the sequence. As I said, the first one, it's AAA. This is the genetic code table. Genetic code table, it's called for the sequence. So first letter is A, second letter is A, third letter. This is the first letter, as you see. This is the first letter is A. Second letter, here. And third letter, here. First letter is A, okay. Second letter is A also, so we have to move to here. Third letter is A, here. A, it's LYS. This is the first amino acid. Second one, it's C, right? C. Let's see if it's pro. First letter is C. Second letter, it's C. And the third letter is C. Here it's C. It's the pro. Let's move now for G. First letter is G. Second letter is G. And the third letter is G. First letter is G. Second letter here, it's a G, and third letter is G, so it's G. Left, G and Y. Fourth one, it's CAA. First letter is C, second letter is A, and the third letter is A, so CAA, GLN. And fourth one, or the fifth one, UCG. So the first letter is U, second letter is C, and the third is G. STR. This is the sequence of the amino acid. Okay? You have to study only 3. three of these tables. This table next year, we're going to talk a lot about this table, how to determine the sequence, amino acids, and so on. But you have to study only three out of these tables. You have to study UAA, UGA, and UAG. These three, they are called stop codon. And whenever we reach in this sequence, you have a stop codon, the sequence of transcription will stop here. There's no more amino acids, so we're going to stop here. This is one of the consequences of mutation. If there is a change in the sequences, for example, UAA and UAG, UGA will code for the stop, and the amino acid sequence will stop here. Okay, this is everything related for the transgenesis process. So as a revision, I'm going to make a quick revision for the transgenesis. Transgenesis is the process where a gene is transferred from one species to another and is expressed in the second species. Transgenic is the organism that can express a gene from another species. And you have to know transcription. It's the moving from DNA to mRNA, and the moving from mRNA to polypeptides called translation. You have to know that the non-template is called the non-transcribed. The coding is the same as the amino acid, the mRNA sequence, but the T is replaced by here, whereas the template strand, the mRNA is complementary to the template, and there is no T. The A is complementary for this, it's good. Okay, so you have to be careful for this sentence. Thank you.